Tarextumab is a monoclonal antibody against Notch, and a randomized phase 2 trial was conducted following an initial phase 1b evaluation of combining Tarextumab with Gemcitabine and Nab-Paclitaxel in patients with untreated metastatic pancreas cancer. And there was pretty compelling preclinical support for this in relevant mouse models of, not mouse models, but in relevant models of pancreas cancer. And that led to a phase 1b evaluation which showed that you could safely combine Tarextumab with Gemcitabine and Nab-Paclitaxel, and we believed we saw an interesting signal in patients with untreated metastatic pancreas cancer, and that led to a much larger randomized phase 2 trial that was powered ultimately for overall survival. And this trial had a co-primary endpoint of looking for outcome in patients with Notch 2/3 high biomarker expression, based on the preclinical data suggesting that this drug may have heightened activity in individuals who had higher levels of the biomarker, and that prognostically they also had a worse outcome. So what we saw, and this was surprising, is that in the overall group there was no difference in outcome for the addition of Tarextumab to Gemcitabine and Nab-Paclitaxel, and if anything, the experimental arm tracked under the control arm. So that was unexpected, not seen during the initial conduct of the study by the Data and Safety Monitoring Committee, but clearly a concern and a worry ultimately. This will not go forward, unfortunately, in pancreas cancer. But we have a lot of potential to learn from failure here, in that patients required on-tissue biopsies, so there is a wealth of opportunity to investigate and try and understand the hypothesis for why the observations that were seen occurred. And so I think more to come on that topic, but sadly the end of that molecule in pancreas cancer.